Okay, so this is then the photosynthesis required practical. And the whole idea of this is to investigate the effect of light intensity on the rate of photosynthesis on an aquatic plant such as pondweed. Now, there's kind of two sort of practical setups that we can do for this. Now, I'll just go through the equipment first. So in this one here, we've got the beaker, we've got a filter funnel, right, which is potentially this here, right? So what it is, it's a filter funnel. We've got a measuring cylinder, which then is over the top. We've got a 10 centimetre piece of pondweed, which is that there. We've got a light source. We've got a meter ruler, and at the bottom there, we've got a stopwatch. There's a stopwatch. And sodium hydrogen carbonate. Now, sodium hydrogen carbonate, the idea of it is that it's going to provide the carbon dioxide for photosynthesis. Now, the way the practical works is put 10 centimetre piece of pondweed, cut edge at the top, into a beaker. Right? So what we've got here is there is our piece of pondweed, and the cut edge is sticking up. It could also be in a boiling tube like that, with the cut edge sticking up. Fill the measuring cylinder with water over the weed. All right, so that there is a full of water measuring cylinder over the top of it. Here you don't have any measuring cylinder, it's just like an open test tube. Then position the light source a metre away from the pondweed. So a metre away from the pondweed. Then start your clock. There's your clock, start it. There's two ways in which you can then count what exactly is then going to be going on. Number one is count and record the number of bubbles released in 3 minutes. So from this little one here, obviously they're coming out of the water, what you do is you count the bubbles. So that one there, count the bubbles coming out of the end of the pondweed. The second one, B, record the volume of gas collected in the measuring cylinder. That is then this one. And what you should see, theoretically, is you should see this filling up with gas. All right. Now, whichever way you're doing it, right, what you're going to do is you're going to kind of adapt it to your results table. Record your results in a table. When you've then done that, you then move the light source so the pondweed beaker is only 80 centimetres away. So then you repeat the experiment, once it's all kind of rested again, to 80 centimetres away. Repeat for then 60, 40 and 20. Then there's a couple of little calculations that then you've also got to do. Right? You need to calculate the light intensity, and you also need to then plot a graph, and I'll run through the graph in a second. One thing to be aware here though is if you use a normal bulb, it will produce a lot of heat, and that heat will then affect the photosynthesis. So best ways around that are use an LED, which doesn't give out heat, or have some sort of plastic in front of it that allows the light through but stops the heat getting through. Right, I'll get onto light intensity in a minute. Right, so variables, don't mess. All right, so dependent is what you measure during the practical. So it could be the volume of gas collected in the measuring cylinder, or it could be the number of bubbles. Independent, change. So what you change between experiments is the distance. The control is then your time that you allow it to adjust, the concentration of sodium hydrogen carbonate, because what you've got to do is you've got to allow the plant to actually adjust to the change in the light. Right, light intensity. Light intensity is proportional to 1 over the distance squared. So an example, I've got an example in that green box there. So it's 1 over, the distance is 2 centimetres squared. So it's 1 over 4, convert it into a decimal, so the light intensity would be 0.25. These are then the sorts of graphs that you might get. You might get asked to do rate of photosynthesis, which is the bubbles of oxygen released, against the light intensity. And what you can do is you can see as the light intensity increases, the rate of photosynthesis increases until it tails off. Or you might get number of bubbles produced per minute, distance from the lamp. right? So the number of bubbles produced is 40 when it's closer to the lamp, and what it then does, it then decreases down. Now, that practical, right, if we're being completely and utterly realistic about it, it really isn't a very good one that works, right? But what you've got to do is you've got to kind of go along. You need to know the method, right? So you've got to be able to talk about the method, and you've got to be able to talk about the variables, right? It's one of those typical ones which is like a traditional write-up of a science experiment. Light intensity is the words that you use, right? You don't use anything other than the light intensity. You don't say light, or you don't say how bright it is. You talk about light intensity. And remember, light intensity is 1 over the distance squared.